action-packed afternoon for invasive species. And for our first talk this afternoon after the break, for something completely different, we're going to be talking about snails. And Chris Pennuto here from Buffalo State is going to tell us all about them.
I'll do my best.
Okay.
First, I appreciate your diehard nature to kind of use something that wasn't strictly about fish. But there are fish involved. So this is some work that one of my former graduate students did primarily, Kira. And the main interest she had here was, in my lab we've been working with gobies for 15 years now on various aspects of their biology and stuff. And she was really interested to see if there were any kind of cascade effects that resulted from interactions between gobies and snails that resulted in changes in leaf decomposition rates. She spoke on this at IAGLR, which was sort of the main thrust of her work, but in part of that was looking at some behavioral things, so I thought we would dig into that a little bit more as a side bar of the work that she did there. So what I'm going to do here, I'm a walker in class, you know. That's kind of weird. The implication here is there's a lot of work that's been done on snail behavioral responses to predators. And certainly one of the take-homes that I want people to remember is that in predator-prey interactions, it's not all about, okay. Got me back there? Thank you. It's not all about predators consume prey, right? A lot of responses in communities result from prey responding to predators, even though they're not consumed. So Schmitz came out with this real nice little dichotomy that you could predict what prey ought to do in the face of predation if you knew the habitat domains of the predators and the prey and the types of hunting modes. And this is his sort of schematic here. And the idea is, again, if we think about snails, and snails might forage throughout the water column on structures anywhere they can find attached paraphyte. So their habitat domain where you can find them is fairly broad. Right? So they're over here, prey broad habitat domain. Predator like a crayfish forages strictly on the bottom. It's not, in the water column. It's not up here. So it has a fairly narrow, where are we at here? A fairly narrow habitat domain for the predator, and it's also a roaming. It roams around. It doesn't sit and wait. So it's an active predator. Thus, when snails encounter crayfish predators in their system, their natural, or the response ought to be some kind of habitat shift to avoid that predator, because they have other parts of the habitat that they normally are found in. Similarly, if we introduce a pelagic or a predator, in this case like a sunfish, a bluegill sunfish, that we can think of as having a fairly large habitat domain, and it also is a sit and wait kind of predator where if you watch these guys forage, right, they kind of hover, look for something, they go after it, they hover, they move around, they're not constantly in motion. So the prediction here is that in the face of a sunfish predator, that snails ought to seek refuge in the bottom, avoiding the pelagic habitat where the sunfish are foraging. And it turns out there's a very large body of literature that suggests and shows that snails do exactly this. You put them in a tank with crayfish, and they all climb up the walls, or they go up the vegetation. You put them in a tank with bluegills, and they all go under the bottom into the leaf litter and stuff like that. Some caveats, different kinds of snails behave or respond a little bit differently, but there does seem to be a large response in this sort of shift. Now, the thing that's interesting, if you're kind of looking in at this, not only do these two predators forage differently, right? Large domains, small domains, but there's something else different about them too. They're actually quite different phylogenetically. One's a fish, one's an arthropod, crustacean, right? So the response of snails, in part, one of the things we want to think about is, well, maybe it's not necessarily the foraging mode of the predator, but maybe it's actually a phylogenetic relationship. Smell fish, do this. Smell crayfish, do that, right? So that's part of what's going to happen in this investigation. All right. Oh, it's a little slow. Okay, so what Kira did, we set two sister experiments. One in the lab, looking at snail response to the scent of different predators, and then a mesocosms design, a study, where we could then sort of try to tease out some of the caramel response, scent responses that we found in a more realistic situation. So in the, for caramones, what we're going to investigate is the response to native predators, fish and crayfish, and to non-native predators, invasive predators, fish and crayfish. So we use round gobies, and red swamp crayfishes are two invasives. And the thing to recognize too, the snails are collected from habitat that is where they're naive to these predators. Neither round gobies nor red swamp crayfish occur in the habitat where the snails are collected, so they've never encountered these before. But the habitat does include the predatory fish and predatory crayfish. So we'll expose the snails to all these different caramones, and then we watch snail behavior through time. So it's a 2-hour trial. They get quantified for their response to the scents every 15 minutes and then every 30 minutes. The way that's set up, it's a small tank, and it's got a 10% caramel solution is administered, and then you begin to watch the snail behavior. And they do some distinct things that we recognize. They crawl up the side of the tank, or in the mesocosms, you'll see they crawl up the vegetation, for example, or the side of the tank. This is called crawl-out behavior, and it's a vertical escape response. They're leaving the benthos. Typically, this is a response you see when they have a benthic predator. Or all the tanks are given some kind of refuge, so they might head for the bottom and seek cover. And this is, again, typical kind of response to a pelagic predator. And then, if they're not under cover or they're not within 2 centimeters of the surface, we just call it non-avoidance behavior. They're just doing what snails do. In the mesocosms, slightly different suite of characters, but again, this was intended to look at the role of gobies in potentially changing leaf litter decomposition. So it's a cascade kind of thing. But some of the same players are here, so I'll use some of this data to show what's going on. So we've got either gobies just with snails, crayfish with snails, crayfish and gobies with snails, and snails by themselves. Eventually, I think, as you'll see, some of the data I'm going to show you is going to focus really on the crayfish for reasons to be seen. That's how the mesocosms were set up. Again, 6 treatments, those different predator combinations with snails, 5 replicates each. And this is set up at our field station at the head of Niagara River, so that we can pump in raw lake water, filter it, get these all set up. It's going to run for 30 days. When they're all set up, a mesocosm looks like this. It's got I don't know if you can see these right here. These are actually artificial plant stems. They're little floating nylon segments of rope. So the snails can climb up those. They climb up the sides of the walls. These prop bricks are also vertical kind of refuge. And then they have, shade cloth and things to keep the temperature. And then they're looked at every 3 days, monitoring the position of snails, in addition to all the other things that Kira was doing. Okay, so two different kind of trials in the lab and then mesocosms. So in the lab, again, these animals are exposed to different caramones, predators they either have never experienced or that they should have experienced. And there's clearly a difference in how the snails responded to the different suite of scents. But a few things to point out. Conspecific alarm cue, right? So each organisms, when they're attacked, their dermis is damaged, their cells are ruptured, they release intraspecific scents. Conspecifics recognize this as an alarm, and they take evasive action, whatever that might be. And they respond consistently and strongly to that kind of a scent. Similarly, largemouth bass or smallmouth bass, native predatory fish that they're quite familiar with from their habitat, the response to that particular scent sustained and fairly strong. They initially respond fairly strongly to gobies, but then they, within 30 minutes, fail to respond appreciably to the presence of round gobies. So that'll come back again. And all the responses to fish are stronger than the responses to crayfish, even though crayfish are quite efficient snail predators. And the difference between the non-native and native crayfish is fairly large in here. And actually, they respond a little bit more to the non-native crayfish. Okay? But again, so this is just a number of snails actually doing some kind of avoidance. If we break it down a little bit, we find out that the snails that are actually avoiding by crawl-out behavior, this particular graph looks very similar to the total avoidance graph we just showed. Maybe a little bit cleaner for some of the things. Again, the conspecific alarm cue is still the dominant response. So looking at, so it looks like all the snails are going up. Very few of the snails go to the bottom. And again, remember, this is the behavior they ought to do with pelagic predators. So in response to the bass, is what they should do. Switch to the mesocosm, and what first thing we notice is the response to gobies was terrible. As you saw in the lab, within 30 minutes they stopped responding to gobies. In the field, all treatments that had gobies consumed all the snails within 2 days. Right? So the gobies are very efficient snail predators. They just ate them all. And then the responses to these two responses where snails did survive happened to be only those treatments where crayfish were present. And when we look at these specific behaviors relative to the crayfish, the two of them crawl out or seek refuge behaviors. What it really looks like in this case is that the snails are doing exactly what the control snails did. Right? So they're the same number of snails in control treatments that are up in sort of our pelagic response behavior are equal to the number of snails that are responding when crayfish are present. So similar to the lab trials, it appears as though they aren't really recognizing the crayfish as significant predators. So, started off with the snails behaving badly. As you now recognize, it's not about switchblades and graffiti. Sometimes they don't respond to predators the way you would expect them to in an adaptive fashion. So they did respond to conspecific alarm cues. That's good. They tended to avoid all predator caramels regardless of the type of predator by the same behavior, and that was crawl-out behavior. Again, you might consider this bad if your predator is a pelagic predator. Again, they responded to non-native predator scents as much as they did to native predator scents, at least in terms of the crayfish. That would be good. But they reduced their response to round gobies fairly rapidly. Within 30 minutes, they stopped avoiding goby scent. Go out to the mesocosms. Again, it could be that failure to avoid for a long term resulted in their demise. So they were actually consumed, like I said, within the first few days, bad. And they also did not even seem to respond to crayfish in those outdoor mesocosm trials, even though those crayfish are actually quite significant snail predators. Again, bad. Okay. In summary, right, a mixture of behaviors that we would anticipate them to do and not, but primarily these snails did not respond the way that we would have thought given the danger of predation. Yeah.
Yeah, just a quick question. So you used bison snails for this study, right? Do you expect that there would be a difference in behavior if you varied the species?